right, hello everybody. So we're gonna finish up chapter 21 lecture. We're looking at concept 3 now. So we're gonna look at evolutions of populations now that we have some background info. So in nature, we know it is not likely that all conditions for Hardy-Weinberg are going to be met because populations are going to be evolving. So what I mean by Hardy-Weinberg conditions are these 5 conditions here. So no mutations, random mating, so you can't select a partner, no natural selection, extremely large population size, and no gene flow is occurring. So if any of these are not met, then evolution is occurring. So we know how easily it is for one of those to not be met. So one of the ways evolution can occur, there's 2 minor mechanisms, which are allele and genotype frequencies due to mutation and non-random mating. So mutations occurring and non-random mating, those are minor mechanisms that contribute to evolution. 3 major mechanisms of evolution are natural selection, genetic drift, and gene flow. So we're going to go into a little bit more info on those 3 major mechanisms. So the first one, natural selection. We've already talked about this. We know it's the differential reproductive success of an organism. So our rock pocket mice, they were able to survive and reproduce depending on the environment they were in. The environment was basically selecting for or against these species, and this was increasing the fitness depending on what color they were. The second one is genetic drift. So what this is an unpredictable fluctuation of alleles from one generation to the next. This occurs, it's going to occur in small populations, so that's where you'll see significant effects. The allele frequencies change at random. So everything with genetic drift is completely random and unpredictable. So you can lose genetic variations in populations due to genetic drift. So this can actually cause harmful alleles to become fixed in a population. So let's say we had some type of genetic disorder that caused a lot of issues. It wasn't good, it was harmful. And let's say that was a recessive allele. So in order to have that trait, you have to have 2 recessive alleles or homozygous recessive genotype. Now, if we had dominant alleles that are in this population, the chances of this harmful genetic genotype popping up are pretty rare. But if something happened where all of those dominant alleles, or most of them, were taken out of this population, now the only people in this species that are mating with each other and producing offspring, they only have the homozygous recessive alleles to give. So that harmful disease is now going to become fixed and stay in this population, which can sometimes lead to potential extinction. So there's 2 types of genetic drift that can occur. It's the founder's effect and the bottleneck effect. So remember, this is unpredictable fluctuations of alleles. So bottleneck effect, this is where we have a severe drop in population size, which then causes certain alleles to be over or underrepresented. So if we look at this picture here, we can see that there was an original population with a really good combination of many different alleles. Some event happened that wiped out most of the population. So the surviving population, we can now see that the blue genotype is very much overrepresented. So like elephant seals, they have reduced genetic variation because people are hunting them. So they have a very small surviving population with very kind of fixed alleles. Same thing with the Florida panthers. They're in danger of extinction because their surviving population and they have alleles that are over and underrepresented. The founder effect is when a few individuals become isolated from a larger population. So this isn't necessarily an event wiping out part of the population, but if a few individuals become isolated, then those alleles will be over or underrepresented in that now new population. So let's say something happened where these little ladybugs were separated from the entire population. It was separation enough where they start mating with each other and forming a new population in this new area. They're only going to have those fixed alleles. So something that is very similar for humans is polydactyly. So this is having 6 fingers. This is very much seen in the Amish population because Amish really only reproduce with other Amish people. So they're not having other alleles come in and um, get rid of this trait. So these individuals basically mate with each other, so we keep seeing those same alleles and phenotypes pop up. All right, concept 4. So we're going to take a little bit closer look at natural selection. We've already talked about it, but this is the true driving force behind evolution. These other random occurrences can happen, and they're a major part of it too, but natural selection is the largest mechanism. So relative fitness is what we look at. This is the contribution an individual makes to the gene pool of the next generation relative to the contribution of others. So if a parent has good fitness and they're able to survive and reproduce, those good genes that they've been able to use in order to survive and make offspring are going to be passed on, which then causes their offspring to most likely have those better or more fit genes. So natural selection, it can occur in 3 different ways. I want you to think of a bell curve as we are going through these next few slides. So one of them is directional selection, disruptive selection, and stabilizing selection. So with a bell curve, in the middle is where your normal or average lies. Anything along the outskirts is considered extremes or considered outside of the norm. So this is what I mean by our bell curve. So if we're using our rock pocket mice as an example, we'll use their color as an example of the phenotypes. So the normal phenotype or something in the middle would be kind of a tannish color. On the very extreme end of the spectrum, we have white, and on the very other extreme end of the spectrum, we have a black color. So with directional selection, if nature had selected for a certain end of the color spectrum, so let's say we had kind of a darkish tan lava flow all over the desert, this is probably where natural selection would place them, where this would be most seen. So we would not have any of the lighter colored rock pocket mice. Disruptive selection, this is typically what we see with our actual rock pocket mice. So what we had seen in the video is disruptive selection. You have a very extreme phenotype because there's basically two different environments, or not environments, but types of environments they're living in. So we have a tan mice because we have our sandy regions. We don't really have a region that's in between because the other sort of place they can be is on dark lava rock. So we see really, really light colored because of the sand, and then we see really, really dark colored because of the lava rock. Now, if there were areas that had kind of a, in between, we would probably start to see an increase in that mid-range color. Stabilizing selection is where we have sort of one basic environment, so we start to see one phenotype rule all of the other phenotypes. So if the desert was just kind of a normal sandy with a mix of lava rock, we would probably see this start to be the dominating phenotype. So balancing selection. Diploidy, this is inheriting 2 alleles, which, as we know, every living thing that sexually reproduces and inherits 2 alleles for every trait, one from mom, one from dad. Now, recessive alleles are hidden in heterozygotes. So remember, that's where they have one dominant allele and one recessive. So there is something that we call a heterozygote advantage, where heterozygous genotypes actually have a better survival rate than homozygous or dominant or homozygous recessive. So a really, really incredible example of this is for sickle cell anemia and malaria. Sickle cell anemia is a recessive disorder. So if you have a homozygous recessive genotype, you are going to die early in your life, probably before reproduction, because your cells are not shaped correctly. If you're homozygous dominant, you are going to have regular normal blood cells. Now, with people that are heterozygotes, they actually have a slight combination of mostly regular blood cells with some sickle. Now, the sickle cells actually help protect against malaria. So, we see in areas that have a high amount of malaria, most people are heterozygous, which means this trait that would normally not be a good thing and you would most likely start to phase out of a gene pool, it actually stays around. That one recessive allele is really, really beneficial for people that live in high malarial areas. Sexual selection. So this is when certain individuals are more likely to obtain mates. We call sexual dimorphism. So this is basically when you can see difference between two sexes. So like size, color, the behaviors. Typically, like we see with male species, they are usually bigger. They have something really flaunty, like lions with their manes, peacocks with the bright feathers. We have deer that have the really large antlers. A lot of the time in birds, they do special dances, or we can see with this, they have kind of the special feathers. So, sexual dimorphism is just seeing differences between two sexes. Now, intersexual selection. This is competition within the same sex. So, and then intersexual selection is mate choice. So, competition within the same sex would be something like females fighting over a male, or males fighting over specific females. Intersexual selection is just choosing mate based off choice. So, because of sexual selection, this can actually lead to pronounced secondary differences between sexes. So, again, this is what causes sexual dimorphism. And the last thing, natural selection cannot fashion perfect organisms. So, natural selection seems like the perfect thing and like it could cause perfect organisms to come about. But as we know, there's a lot of stuff with every single species that isn't perfect. So, the reason for this is selection can only edit existing variations. So, if we have not had a mutation or variation occur, it can't act on it. Evolution is limited by historical constraints. Adaptations are often compromises. So, adaptations, as we know, it can be beneficial in one area and not in another. And if that environment changes, it is no longer an adaptation. And the most important one, chance, natural selection, and the environment are all interacting together. So, if natural selection was the one driving force, then we could potentially start to see more perfect organisms. But because we have things like chance, where we have bottleneck effects and founders effects and genetic drift, and we also have environmental changes that are random, this is never going to fashion perfect organisms. So that's it for chapter 21. Let me know if you have any questions. 7.6 video questions are your homework for tonight, and those are going to be due tomorrow. Do not forget 7.5 video questions are due tonight as well. You will also have a chapter 20 quiz tomorrow. So all of this information we just covered is not on that. It's going to be strictly on phylogeny. All right, have a good day.